Alrighty. Aloha, everyone. Welcome to this week's TCBES Research Seminar Series. I have the pleasure of introducing Dr. Matthew Knope, who is presenting the research projects of 4 recent TCBES graduates, their findings and challenges from the ecology of Hawaiian plants, fungi, algae, and invertebrates to global assessments of extinction risk invertebrates. He received a Bachelor's of Arts degree in marine biology from the University of California at Santa Cruz, a Master's of Arts degree in marine biology from San Francisco State University, and a PhD in biology from Stanford University. Dr. Knope is now an associate professor in the Department of Biology at UH Hilo and runs the Knope Evolutionary Ecology Lab. His research interests are in ecology, evolution, and conservation biology, specializing in the speciation and extinction of plants and animals in Hawaii and around the world. The main goals of the research at his lab are to integrate the way biologists study living organisms with the ways fossil organisms are studied, and to use genomic sequencing technologies to unravel the evolutionary history of Hawaiian plants and to aid in their conservation. With that, I would like to welcome Dr. Matthew Knope.
Olivia, thank you very much for that warm and generous introduction. And thank you all very much for being here. I really appreciate it. Yeah, let's just go ahead and jump right in. Let's see. I'm going to advance the slide. Yeah, the first thing I want to do is just start with acknowledgements. And there are so many people to thank. And I've listed some here, and I've undoubtedly missed many. But I want to thank all of you, everybody who's involved in TCBES. It really is, as you all know, it's a team effort, right? It's just been extraordinary these last 6, now going on 7 years to be a part of TCBES. I also want to thank everybody in my lab. There's a couple of pictures here of folks in my lab. You may know some of them or all of them who have just been wonderful to work with. And I've been very fortunate in that regard and how the lab has developed and the people who've been attracted to work in it. And I want to thank funding sources. The Department of Biology at UH Hilo, Stanford University, NSF, and UH Hilo have all been very generous, and the financial resources that make this work possible that I'll talk about today. Okay, so first, I just want to take a step back, and I want you guys all to absorb these photographs, many of which are my own, and just take a moment to just remember this extraordinary biodiversity that we share this planet with, right? And this is really ultimately, what gets me up and excited every day. And as Olivia said so well, my lab's really interested in kind of the interface between ecology, evolution, and conservation biology, and we tend to gravitate towards questions that revolve around the forces that generate biodiversity, maintain biodiversity, and ultimately destroy biodiversity and lead to extinction. And we're going to talk about some of all of that today. On that note, what I'd like to do with this seminar, this is a brand new seminar, I've never given it before, so hopefully it goes well, and I look forward to any feedback, constructive or otherwise. As I did some Q&A with 4 recent TCBES graduates that have worked in my lab. You may or may not know them. They've all graduated, some just last semester and some a little more distantly. But Becca Ward, Jeff Stallman, Maya Munsterman, and Trevor Bach. And what I want to do today is kind of twofold. I want to talk about their research itself and the findings, and maybe what we feel are some of the important things about the findings of the research. But I also want to talk about their research process and their time in TCBES, and in particular challenges and triumphs. And so I asked them all the same series of 6 questions, and I'm going to kind of sprinkle those throughout the talk as I move through each of the 4 of them and their research projects. And I'm going to try and make the talk a little bit interactive and get your guys, kind of thoughts, feelings, experiences about research and about challenges and triumphs, and ultimately maybe, collectively between the wisdom of these 4 recent graduates and the rest of all of you, we might find there's some commonality and that some things really resonate, as the whole group in terms of, what is this endeavor that we're doing, and, how do we move forward and support each other the best that we can. So having said that, here's the first audience participation. I want everybody to open their chat. And can you answer this question in a simple yes or no? Have you experienced significant setbacks in your research? And hopefully everybody can write in. Yeah. Yes. All yeses so far. Yes, 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 yes. So that's exactly what I thought. And there's not a single no, right? There's not research that we do that doesn't have significant setbacks and significant roadblocks. That's just part of the process. But then it becomes, okay, well, how do we deal with that? And hopefully, yeah, through this seminar, we'll start to, like I say, kind of come up with some things that are common themes for kind of ways to help ourselves and help each other. Now, though, before I move on, the second part of this first audience participation is in 3 words or less, not more than 3. How have you overcome or are currently working towards overcoming those setbacks? Give it a second and then write 1, 2, or 3 words, but not 4 or more. How are you working on overcoming those setbacks or have already overcome those setbacks? A lot of hope, yeah. Collect more samples, communication. This is great. Pivoted focus. Yeah, Lisa, that's an important one that really resonates with me. Communication, trial and error, patience again, asking others, more background research, again, more research, reframing perspectives, again, pivoting, resetting, writing a lot. Okay, this is excellent. So this is exactly what I thought. And there's definitely, as you can see, there's some common themes that are coming up here. It's not all the same, but there's a few things that you're all kind of sharing in common about your experience and moving through your research projects. Keep them coming if you haven't had a chance and you're still thinking about it. But I'm going to go ahead and move the next slide just for the sake of time. So these next 2 slides are going to be kind of text heavy, but then we'll get back to some pretty pictures and some cool figures and stuff in a minute. But I wanted to start with these 2 first questions that I asked Becca, Jeff, Maya, and Trevor and give their responses in order. I asked them what was the most challenging aspect or aspects of their research while they were a TCBS student. And Becca said, for me, there was a big learning curve of learning to trust myself and take charge of my research, even more than learning statistics, project management, and working with collaborators, all of which are challenges in themselves. Jeff said stats, although that is still a challenge, learning when to ask for help versus tough it out and hoping an issue will resolve with more work expertise through practice, and also the work-life balance. Maya said one of the most challenging parts of TCBES program was managing all the responsibilities of research, mentoring, coursework, and external jobs. And Trevor said, really, COVID outweighed all the other challenges. COVID was such, let me just move my chat so I can read it here. A dramatic shift in day-to-day life, was very damaging the social life and social support network. And this seems to be true across all schools based on what PhD students and labs I'm interested in have told me. And for me, at least, really decreased my motivation to work and to finish my thesis the more COVID dragged on. How many people does that resonate with? Right, yeah. Okay, so then I asked them, how did you ultimately overcome these challenges? And Becca said, I think overcoming this takes time. This is learning to trust herself. I think overcoming this takes time and is part of the journey of grad school. As you invest hours into developing a baseline of mastery in your research topic, and as you brainstorm and collaborate with others, and as you refine your project, you learn to trust yourself more. Whoops, sorry. Jeff said, talking to others in the program and faculty and advisors, recognizing that long-term research projects and advanced degrees take long periods of time. While I may have had to sacrifice some things to make time for grad school related things, I always needed to make time for myself and non-school related activities, because in the end, it's 2 to 3 years of your life, and you can't put the rest of your life on hold for that long, nor do you want to. The project may still take a full 2 years or 3 years, in my case, Jeff is saying, whether you work on weekends or stay up late every night working on it or not. And Maya said, time management. There was always more work to be done, but allotting time to unwind is also super important. I always tried to keep the end goal in mind, and for her, graduating in 2 years was important when doing my research. This helped me stay on task and manage my responsibilities efficiently. And Trevor said, at the end of the day, perseverance is the main thing that allowed me to overcome COVID, and a commitment to my goal that I came out here to complete a thesis and advance my academic journey. And I was still determined to do that. Working with the co-authors did really help as well for me, as you predicted. It was very motivating to work with such high caliber scientists and see how they work through problems. And as I got more into working with them, I didn't want to let them down by not finishing or not publishing. So that helped give me an extra push as well. Okay, so I know that was very text heavy, but I wanted to kind of give you that first, that, you know, these were the things that were their biggest challenges, and these are the answers for really how they felt they overcame those challenges. And I think there's really important insights in all of that. Okay, now I want to kind of jump into individual research projects, and then I'll talk a little bit more about where kind of each Becca, Jeff, Maya, and Trevor are now, and where they're going, and a little bit about their advice for all of us moving forward. So Becca is currently the resource coordinator for the pre-K-12 Research and Development Consortium. And that's essentially what this question, I asked each of them this question: what's your current position title, and what research, education, service, et cetera, you're currently working on? And she says, I'm the research coordinator for the Hawaii Pre-K-12 Research and Development Consortium, the RDC. Through this position, I work collaboratively and remotely with the Akaka Foundation for Tropical Forests to help the RDC achieve its objectives of fostering reciprocal relationships between the science community, indigenous peoples, and the K-12 education system. In particular, I support the development of Hawaii-based educational materials rooted in local ecological history and indigenous knowledge. Okay, so when Maya came to UH Hilo and the TCBES, Becca came to UH Hilo and the TCBES program, she was really interested in climate change and the possible impacts of warming sea surface temperatures on marine communities. And ultimately, we developed a project looking at the possibility of changing sea surface temperatures in the main Hawaiian Islands and how this may be impacting intertidal ecological communities, in particular the limu and the invertebrates that live in these communities. And so if you just quickly, take a glance at the figure on the left, this is probably, something you've seen before or similar to things you've seen before. These are global temperature anomalies from 1891 to 2020. This is data compiled by the Tokyo Climate Center. And it's alarming, and it's perfectly clear. The world is warming, and it's warming rapidly. In Hawaii, one particularly influential model predicts that the rate of warming is expected to increase by 250% by the end of this century, by the end of the 21st century. And that will bring us to about 0.4 of a degree per decade for the rate of warming, which is about 0.7 of a degree Fahrenheit, 4/10 Celsius or 0.7 Fahrenheit. And that this has the potential for widespread consequences for marine ecosystems. And of course, warming waters can be an advantage to some species or could be a disadvantage to some species, right? And so these are the kinds of questions that Becca was really interested in. So she assembled an amazing team of 2 undergraduates and a high school student. Noelle Haddad had finished her degree at San Francisco State in marine biology and joined the research team. Cassie McTee was a national student exchange student from Rutgers that came to UH Hilo, loved it so much she decided not to go back to Rutgers and transferred and finished her degree in the biology department at UH Hilo. And Megan Nakamoto she's now graduated, a high school student at Waikea and she's worked both with Maya's project, I'll talk about it in a minute, as well as Becca's project over the course of 2 years, and is just a phenomenal, very high potential young scientist. So we teamed up with OPE, or Our Project in Hawaii's Intertidal. Everything in biology, everything in science is an acronym, right? And this is one of these kind of clever ones, right? OPE is the Hawaiian name for the limpet, common intertidal gastropod. Many of you I know this. But Our Project in Hawaii's Intertidal is a citizen science project. It's basically pairing middle school and high school students throughout the state and their teachers with professional marine scientists, both at UH Hilo and UH Manoa. And in total, there's something like 42 sites now throughout the main Hawaiian Islands. It's become a very successful program. And it started in 2005. I got involved myself as a graduate student in 2006. There was a hiatus of about 8 years, when funding dried up for a little while, but it's back strong, and it has been for a while. There's been published research that's shown that it's very high quality data, that the citizen science data is almost identical to what independent surveys from professional marine scientists of these same intertidal sites throughout the islands come up with, especially for the more abundant, easier to identify species. Some of the real cryptic, rare species, that's the kind of less reliable parts of the data, and so we avoided focusing on those. I don't have time for details of the monitoring methods, but if you're familiar with basic transects and quadrats, subsampling of an ecological community, and it's uniform throughout all of the sites. There were 12 sites in particular throughout 4 of the main Hawaiian Islands that had the most data over time, repeated measures from the same sites to focus on, and those are the sites that Becca ultimately ended up analyzing. For sea surface temperature, she worked with Dr. Stephen Colbert in the Marine Science Department and downloaded satellite remote sensed sea surface temperature data from what's called the OSTIA, the Operational Sea Surface Temperature and Sea Ice Analysis Satellite Data Center, something run by Copernicus. This has a resolution of about 6 square kilometers, so that's like the kind of level of resolution for the data in terms of monitoring daily sea surface temperatures throughout the main Hawaiian Islands and at the nearshore 6-kilometer grid adjacent to these intertidal sites. She looked both at the near real-time data as well as the reprocessed data, and they're almost identical. They're statistically indistinguishable, and she analyzed both. She did this both for each of the 12 sites and for the average daily sea surface temperatures for the main Hawaiian Islands, and I'm going to show you data from both now. So this is the kind of whole enchilada across the main Hawaiian Islands from 2004 to 2019 on the x-axis here. We're seeing time on the x-axis and sea surface temperature in degrees Celsius. And overall here, what you see is that the average sea surface temperatures in Hawaii across the main Hawaiian Islands are increasing. And they're increasing at about 0.4 degrees Celsius per decade, which is just about exactly identical to what this influential model has predicted for end of the 21st century. So basically, this 15-year interval that we just went through had a rate of warming that we were not expected to see for another 80 years. The other thing that's really kind of alarming about this data is it's not only that the mean temperature is increasing over time, but the yearly maximum temperature. These peaks are the summer highs, and the summer highs across the main Hawaiian Islands in the second half of this 15-year interval are significantly greater than they were in the first half. So it's not only that the mean's going up, but also those real peak temperatures are going up as well. This figure just blows me away. This figure is again time from 2004 to 2019, and again sea surface temperature. The black line is the main Hawaiian Islands as a whole, and each of the colored lines are each of the 12 intertidal sites independently. And here's the color legend. This is it just totally blew me away because the slopes of these lines are almost identical to each other. All 12 sites are increasing in temperature at almost the exact same rate. They're starting at largely different temperatures and they're ending at largely different temperatures, but they're almost exactly mirroring each other, as well as the picture for the entire island chain, at least the main Hawaiian Islands. Okay, now in terms of the ecology and coupling this, increased sea surface temperature to what's happening in the intertidal for limu and invertebrates, I wish I had an hour to give a whole seminar about it. I wish I had an hour to let Becca explain it, but I'm just going to give you some highlights that I think are particularly important. First, I'll ask you to focus on this kind of upper left figure, which is the turf algae. And turf algae is a kind of catch-all where it's difficult to identify taxonomically to the species level. So we, with Opihi, identify that as a kind of conglomerate of turf algae. And you're probably familiar with this if you've been out in the intertidal. The thing that you're seeing here is a statistically significant relationship with increasing sea surface temperature leads to a dramatic decrease in abundance of turf algae across these 12 sites. And the window of time where those temperatures are really important is about 3 months to 11 months prior to the ecological sampling. So the warm water that's really important for determining the abundance of turf algae is not what happened yesterday. It's not what happened last week. It's not what happened last month. It's what happened 3 months to almost a year ago. Does that make sense? Similarly, Padina has a very similar kind of time window of 3 to 12 months, but the relationship is the reverse. Padina increases in abundance dramatically, and both of these are important ecological players in intertidal communities. And you can see really dramatic increases in Padina that's coupled to these increasing temperatures. Another really interesting one that I want to highlight here is the invasive Acanthophorus spicifera. So this is a highly invasive alga at certain intertidal sites. Unfortunately, it's just shown up, for example, there in Keaukaha at some of the sites near Hilo. And it has, again, a statistically significant and positive relationship with these warming temperatures. So this isn't good news, right? Not only that it's spreading, but that the warmer waters are creating conditions that seem to lead to its increase in abundance. I don't have time to get into the other ones now, but you get the point here that the response of individual organisms to the sea surface the rising sea surface temperature is varied. But that's ultimately leading to change in these ecological communities. So I think what we can say safely is that sea surface temperatures across the main Hawaiian Islands warmed at a rate near what's predicted for 80 years from now. Right? This 0.4 degrees Celsius per decade isn't the end of 21st century. It's happening right now. The second half of the study's time frame also had significantly greater maximum yearly sea surface temperature. So those summer highs are just getting hotter and hotter. And that these warming sea surface temperatures appear to be influencing Hawaiian intertidal communities. And I don't have time to show you all of the gory details, but I'll tell you that the take home is it seems to be driving these communities to be more dissimilar from one another. So they're starting closer to each other ecologically, and they're moving out in different directions as the waters are warming. Okay, so I asked Becca, where's she going from here? What are your career goals, and how has the TCBES program contributed or not to you obtaining those goals? And she said, I would like to continue working in spaces that empower community-driven stewardship, that uplifts Indigenous knowledge, and foster connections between people and the lands they inhabit. I think the TCBES program was an incredible opportunity for me to develop really valuable skills that I bring into my work every day, whether it's critical thinking, collaborative skills, or I think it says project management, and to move the tiles there. That was Becca's project. Now I want to shift gears and talk a little bit about the work that Jeff has been doing. And Jeff was kind of Hanai in the lab, in that he was officially advised by Mike Shintaku and Don Hemis, and did a whole separate master's thesis with them that they published. I think they've published a couple of papers out of it, but he and I hit it off, and we did a couple of projects both on native Hawaiian plants as well as native and non-native mushroom species, and I'll get to that in just a minute. Okay, so he's currently a PhD candidate in Dr. Mary Catherine Amy's lab at Purdue University, and he says, I'm currently working on the taxonomy and systematics of the fungal class Leodiomycetes, with a focus on tropical macrofungi, more simply, cute cup fungi from warm places in this class. And I'm still trying I move the tiles here again. I'm still trying to pursue research on Hawaiian fungal biogeography, conservation, and biodiversity. And he's being very humble there. He's doing amazing work in that avenue. Okay. So the first project I want to talk about is in relation to native flowering plants. You are probably likely aware there's about 1,000 species of native flowering plants in Hawaii, and they've all descended from just over 250 original colonists. This is work by Dr. Jonathan Price of TCBES and the Geography and Environmental Science Department with Warren Wagner at the Smithsonian. And it's, again, take a step back and absorb these pictures. I mean, it just blows me away. These are the organisms that we get to work with. It's an honor and a privilege to work on these plants. However, these plants, many of them are highly imperiled. Before I get into those details, I want to just talk about what I like to call the Hawaiian plant paradox, which is that in many of these lineages, we have often extreme ecological and morphological differentiation. Think about the silver swords, think about the lobeliads, think about the Bidens or the Ko'oko'olau. It can be so different from each other ecologically and morphologically, but often they have very limited genetic differentiation. Every one of these citations that I've given on these pictures are all published studies that show extremely limited genetic differentiation amongst these otherwise widely divergent species. So DNA barcoding, you may or may not be familiar with, has been largely successful in differentiating animal species, including animal species in Hawaii. For example, the native Hawaiian bee and the genus Hylaeus has been published shown to be pretty effective, about 95% successful to use DNA barcodes. But for plants, it's been trickier than it has been for animals. And that's probably a statement you could just say generally about differences between research in plants and animals. So the most effective loci, that is the genetic loci, and the evaluative methods for plants are still hotly debated. And floras of young oceanic islands like Hawaii and others are a challenging test of DNA barcodes because of the often recent and rapid speciation and the high incidence of hybridization and polyploidy. So I think the barcoding is an unfortunate name, but it seems to have stuck. But basically the idea, if you're not familiar, is that the DNA sequences, a small snippet of DNA can essentially serve like a barcode, on a box of cereal, and you go, to Safeway or KTA or wherever you do your shopping. And that barcode at the cash register is what's going to tell the computer whether or not it's Wheaties or it's Cheerios or it's, Great Grains granola or whatever, and charge you hopefully appropriately for it. The same idea is that this little snippet of DNA should be unique to each species. And so they've then, called these DNA barcodes. So this may be helpful in plant conservation in Hawaii, where there's over 1,000 native species. As I just said, it's hard for botanists to know every single one of them. About 10% of the native flora is already extinct, and more than half of them are considered at risk, that is vulnerable or worse. And more than 200 species have fewer than 50 individuals left in the wild. That's, it's an alarming statistic, right? That many of these plant species are highly, highly, highly endangered. So DNA barcoding has the potential to provide a more rapid identification of species with a small piece of tissue without the requirement of a trained taxonomist examining a mature flowering plant, which is generally what you need for a confident diagnosis, right? And the sequencing technology has now been taken into the field. And so, for example, it's happening here. This is a handheld DNA sequencer in the rainforest in Borneo. And this researcher is simply putting a little tissue, a tiny little bit of leaf, the smaller the amount, the better. And it's being read real time by this DNA sequencer. It's being pinged to a satellite. It's being run through a barcoding database. And the answer is coming back within minutes to what species. And in rainforests in Hawaii and Borneo and other tropical regions of the world, there can be thousands of species in a single forest, at least in some places in the world. And so there's this potential and there's this excitement that this could make it so that we can really identify lots of species very rapidly, including what are the endangered, critically endangered ones, even when diagnostic features are not available to us. So Jeff set out to test this in Hawaiian plants, and he tested 385 native Hawaiian plant species in 20 different evolutionary lineages. And he used all 4 of the commonly proposed barcodes for plants, which is ITS, TurnH, MACK, and RBCL, and also what's called the concatenated set, which is basically putting together all 4 of these loci. He also used the 3 most common evaluative methods, that is a character-based method, a DNA character-based method, a phylogenetic tree-based method, and a genetic distance-based method. And here is the percentage identified across all of these different methods, across all these different loci in the concatenated set. And at best, he's able to get 22% success for each, of these 20 evolutionary lineages. Basically, DNA barcodes just fail miserably for Hawaiian plants, and he was able to publish these results recently. So question becomes, should we just give up? What do we do and where do we go from here? And I don't think we give up just yet. And there is some kind of promising technologies on the genetic horizon. We've done whole plastome sequencing, that is the chloroplast genomes that are primarily maternally inherited for bidens or ko'oko'olau, and they seem to be really good at species level diagnosis. Others like Henrique Krenwinkel working in Kipuka on Hawaii Island have used those handheld nanopore sequencers that I just showed you from the forest in Borneo, and have had promising results for plants and arthropods using these long read ribosomal DNA sequences. And then recent approaches that are essentially reduced representation genomics that go after large numbers of, for example, 353 loci, that's a kind of angiosperm wide set of loci, or for composites over 1,000 loci. These may be where we're going, right? And in 5-10 years, this is going to be cheap, easy stuff. It's not exactly right now, but it will be, mark my words. So I think that's probably where we're going with plants, is to kind of more data. So switch gears a little bit now and just briefly mention this new paper that we have in press. It should be online, open access any day now. Very excited. This is work that Jeff did, teamed up with an undergraduate in the biology department, Kira Robinson, who also has really strong interest in fungal biology. And together we worked on this project, basically asking the question, do endemic mushrooms on oceanic islands and archipelagos, including Hawaii, support the theory of island biogeography? And I just want to show you one figure, and then I'll tell you about some more results just for the sake of time. But Hawaii here is set up for percent endemism on the y-axis against the Galapagos, the Canary Islands, Cabo Verde, Madeira, the Azores, and Christmas Island for Agaricomycetes. These are the mushroom-forming fungi. Lichens in this kind of orange color, the Agaricomycetes are this kind of teal color, ferns in this blue, angiosperms in pink, and bryophytes, right? These are the mosses, hornworts, and liverworts in green. And one thing that you'll see right away, right, is Hawaii is exceptional. The levels of endemism that we have, pretty much across the board is really high. This, though, is amazing, right? That mushrooms in Hawaii have almost the highest percentage of endemism as flowering plants, right? Exceptionally high, and way higher than any other island or archipelago that we had comparable data for. So Hawaii has the highest mushroom endemicity at 83% of these islands and archipelagos. And it looks like about 50% are potentially derived from what's called in situ cladogenesis. That is speciation that's happening within the archipelago. And we find positive and statistically significant relationships between the distance to the mainland and the island area with percent endemism among mushrooms. So the further away you are and the bigger you are, like Hawaii, the higher your percentage of endemic mushroom species. And that cladogenesis, that is speciation in situ, is positively and significantly correlated with the size of the island and the elevation of the island. So big islands like Hawaii Island that are both big and tall tend to have more speciation events than small islands that are low-lying. Probably not a surprise. But together, collectively, these results are at least preliminary evidence that endemic mushrooms support the predictions, the general predictions of the theory of island biogeography. So we're really excited to get these results out there. So then I asked Jeff, what are your career goals and how has the TCBES program contributed or not to obtaining those? And he said, I'm still not completely sure. And probably many of you can relate to this. My dream job would be a curator of an herbarium or a fungarium, although these positions are hard to come by. I would be interested in being a professor at a non-R1. This means a university where research is not the first focus, but teaching is the first focus. Working for a federal agency such as the USDA or the USGS, doing research or doing natural resource land management or public-private nonprofit organization. So he's still pretty open in terms of direction here, which is probably, on some level, a good thing. For me, not having an academic background in environmental sciences and conservation biology, Jeff did his undergraduate work at the University of Washington in Russian literature. But having a bit of work experience and a passion for the topic, TCBES was a great stepping stone to getting a better job in the islands and helping me determine if I liked research and studying fungi on an academic level, as I did in my free time and as a hobby for many years. Because I didn't have a specific career goal back then, it being a small program with a lot of variety of projects, it exposed me to a lot of different research topics and techniques. Okay, so now I want to talk a little bit about Maya's work. And Maya's work and Trevor's work are both conceptually and methodologically very closely related to each other. So I'm going to kind of set up the methods with Maya's, and then I'm going to fly through them with Trevor's and kind of cut to the results more quickly in Trevor's. So Maya, I asked her what she's up to now, and she's currently also a PhD student. She's at Florida State University working with Dr. Dan Okamoto as her advisor, but she's physically located at the UC Davis Bodega Marine Lab in Northern California, researching the effects of ocean warming and marine heat waves on purple sea urchin populations and their broader implications for kelp forest ecosystem health. So I want to ask a question, and again, I want audience participation. So everybody open their chat again if you closed it, and I want to ask this question: is the sixth mass extinction, and this is in reference to the previous big five you've, I'm sure, heard of, currently underway? Yes, no, or maybe? If everybody could vote, that would be great. Yes, no, or maybe. Are we in Earth's sixth mass extinction now? A lot of yeses, a couple maybes, a few more yeses, a maybe. Becca's abstaining because she knows my answer. She's heard me give this before. I like to ask this question. And a couple more maybes. Okay, this is great. Wonderful. So a lot of yeses, a lot of maybes, no nos, right? Everybody recognizes that we're in a serious crisis, right? Does it rise to the level of a mass extinction? Well, let's take a look at some of the data. So one important study that came out about a decade ago by Tony Barnosky and colleagues looked class by class at the percentage of species that have already gone extinct in white here in the current biodiversity crisis. So you can see here, for example, mammals, about 1%. Birds, about 1%, reptiles, about 1%. The asterisk is saying there's some data uncertainty, amphibs, about 1%, et cetera. Scleractinian corals, basically zero documented extinctions. Actinopterygii, this is the ray-finned fishes, which are the vast diversity of fishes. And this is important for Trevor's work. This is what he focused on. I'm going to come back to that. Documented maybe about 1%. The numbers here in black are the percentages that if all currently threatened species were to go extinct, every one of them were to go extinct, about 20, 30, in some cases, maybe up to 50. For cycads, 64%. Think about that for a second. 64% of all cycad species are threatened or worse. But none of these groups, not a single one of these Linnaean classes, would rise to the level of the previous big five mass extinctions, where it's thought that 75% or more of all species went extinct. I will mention, though, that this statistic gets thrown around a lot, and that most paleontologists kind of bristle at this, that it's not well constrained at the species level. What is well constrained is that about 50% of genera have gone extinct in some of these mass extinctions. But whether or not it's 75% or more, that's something that there's still considerable debate about. But the point here is that even if all threatened species were to go extinct, we're not in a mass extinction in terms of magnitude. But magnitude is not all there is to know about any extinction event. There's two other critically important pieces that I want to tell you about today. One is rate, how fast are species going extinct? And so magnitude, the second I should say, is rate. And then the third is what's called selectivity, which types of species are going extinct. So let's take a look at rate. And this is the rate for terrestrial vertebrates, which, as you guys all know, are by far the most well-studied group of organisms on the planet. That's just the reality, right? And if we take the rate at which terrestrial vertebrates are currently being driven to extinction and project those rates forward, which was what Bartnoski and colleagues did, it'll take about 250 to maybe as much as 2,300 years to get us firmly into the sixth mass extinction in terms of magnitude, to get to this 75% threshold, right? So couple of centuries, a couple of millennia at most to get there in terms of magnitude based on rate for terrestrial vertebrates. Now, in terms of selectivity, one of the things that is very clear about the current biodiversity crisis is that species aren't equally at risk from one another, right? And this is really abundantly clear when you look at terrestrial mammals, right? What humans did in the Pleistocene is they drove big animals extinct first. In this case, it's terrestrial mammals. So you think about, these giant sloths, cave bears, saber-toothed cats, et cetera, et cetera. And basically that trend has continued. And the things that are not threatened in a modern world are things that are small, and things that are being driven extinct are things that are progressively larger. So this is what we call strong ecological selectivity, or in this case, body size selectivity as the ecological trait. So this is what Maya got really interested in. And what she and her team did is they used the published literature to assign 3 core ecological traits. That is the habitat association, the mode of locomotion, and the feeding mode for roughly 18,000 species of terrestrial vertebrates for which they could find published data, and matched these species to their IUCN Red List category of extinction risk. And for those of you who aren't familiar, this is basically how the IUCN categorizes extinction risk. Species that are least concern to those that are near threatened are generally grouped together as non-threatened, whereas things that are vulnerable or worse are considered threatened. And she then applied large-scale species-level molecular phylogenies in logistic regression analyses with this kind of binary extinction risk, threatened or non-threatened, as a response variable, and her ecological traits as the categorical predictor variables. The reason she used these phylogenies was to account for any potential phylogenetic covariance in the ecological traits. And lastly, she examined the individual categories of extinction drivers that are threatening each species, and she determined the greatest threat for each of the 4 terrestrial vertebrate groups. She also then quantified the sum of the risk factors, the different number of threats that are threatening each of these species, to provide a multi-stressor perspective on threat. And as I mentioned, these methods are very similar to what Trevor did. So when I get to Trevor's part in the last part of the talk, this is basically what he did as well. So Maya recently published these results, and she published them not only in Conservation Biology, of which is an academic journal, but she also published these results in a popular science magazine that's called Current Conservation, to translate these results to the general public around the world. She also used her research and brought it into my undergraduate evolution class, and we did some pedagogical study to basically determine kind of what methods of bringing authentic research into undergraduate classroom work most effectively. So she's been a very productive scientist. So in terms of her research, what did she find? She found, and these first results are not novel. These are things that the scientific community has known, but I just want to highlight that basically her results support what others have found, which is that amphibians are highly threatened. Greater than 40% of all amphibian species in this data set of 18,000 species are threatened or worse. Reptiles, mammals, and birds, none of them are doing great. Overall, about 25% of all terrestrial vertebrates are threatened with extinction. This is novel, though. What she did is she tallied the threat types for each species that are listed for these species that have been assessed by the IUCN. What's driving these species to be threatened? And she found that the #1 driver across all terrestrial vertebrates is commercial agriculture. then logging, then invasive species, and then development as the top 4. And I don't have time to show you the results, but I'm going to tell you that each of the 4 Linnaean classes, amphibians, reptiles, mammals, and birds, all also have those same 4 in the same rank order, the same top 4. Beyond that, they tend to vary in terms of, the rank order of the threat types. I want to point out that climate change currently ranks relatively low as a threat type for terrestrial vertebrates, but that we expect that this will unfortunately likely change as the climate crisis continues to accelerate and intensify. And of course, these threat drivers are not independent of one another, right? So I don't think there's much solace in its placement as a relatively low threat driver at this point. But one thing that's just alarming is that commercial agriculture is now a dominant human influence on the planet. It's now covering approximately 40% of the world's ice-free land surface. Think about that. 40% of the world's ice-free land surface is devoted to commercial agriculture. So maybe it's no surprise that this is the #1 threat type for terrestrial vertebrates globally. In terms of the ecology and the ecological categories that are most at risk, I just want to, this is all busy, I'm just going to highlight a couple of things. It's cave-dwelling amphibians, more than 75% are threatened with extinction. It's arboreal quadrupedal, that's a fancy name for mammals that swing, brachiating through forests, are also almost 80% threatened, right? This is primates that are, using kind of swinging locomotion in forests. And scavenging birds are really, as ecological groups, experiencing disproportionate risk and warrant immediate attention and protection. And in this paper, we argue, before I do that, I'll just say that this part here is showing the number, as I said, we tallied up the number of threats that threatening each of these species. And what we find is that for all terrestrial vertebrates, for amphibians, for reptiles, and for birds, the species that are in these higher endangerment categories, endangered or critically endangered, versus near threatened or vulnerable, have significantly greater number of total threats that they're facing. That's not true for mammals. It just seems to be about the same. But we essentially, across all species here, the signal is pretty clear. It's a death by 1,000 cut scenario. So basically, a species can handle a threat or 2, but it can't handle 4, 5, 6 at the same time, right? It's just like us. More things that are attacking us at the same time, it just becomes too much. And then we argue in this paper that a shift in focus from a species-centric to a trait-centric conservation practice will allow for protection of these at-risk functional diversity categories from both regional to global scales. Okay, so then I asked Maya a bit about her career goals. And she said that her short-term goal is to finish her PhD and obtain a postdoc in marine ecology. I have to move the chat now, and global change biology. Her long-term goals are either to become a professor or a museum curator using genomics to research the effects of climate change on marine ecosystems. Excuse me. TCBES provided me with the introductions to research. I learned how to code in R. I managed a team of undergraduate students, and I forgot to highlight that before, but she had just a stellar team of undergraduate students that worked with her for, I think, all 4 semesters. And to collaborate with colleagues from other universities, submit a manuscript for peer-reviewed publication, and to present at both national and international conferences, and engage with the local community and culture and explore research or opportunities outside of my master's study. I have used each of these skills many times since graduating from the TCBES program, and I would not be where I am today without the experiences and relationships I made during my time at UH-Hilo. Okay. So last, but certainly not least, I want to talk about Trevor's work, and I have just about 10 minutes to do it here, so I think it's about perfect. So Trevor currently is employed as a quantitative technician for the Research Corporation at the University of Hawaii. And he works as part of the Pacific Islands Ecosystem Research Cooperative. I think this is cooperative. Maybe somebody can correct me if it's not. With USGS. I primarily use statistical models in R to estimate bird population levels around the Pacific using field data. And he's also working on a project that is seeking to estimate ungulate densities using camera trap data in Guam with a largely automated workflow. Pretty cool stuff. So in terms of the extinction crisis in the oceans, one really influential paper came from Doug Macaulay and colleagues, published in 2014. And one of the things that's perfectly clear about the signal here is that marine organisms that come into contact with land, so sea turtles, pinnipeds, and otters, seabirds and shorebirds, sea snakes and lizards, are at high levels of endangerment. That's a very clear signal that if you're coming onto land as a marine organism, you're basically in big, big trouble, right? And that probably doesn't surprise anybody. At first glance, you might think, oh, hey, there's not a lot of red for things that are exclusively aquatic. But if we look at this more closely, the dark gray is unreviewed. The light gray is data deficient, right? So even though there's not a lot of red and a lot of yellow, which are extinct or endangered, it's likely much more due to the fact that we just hadn't, as of 2014, had enough data to really get a picture for exclusively marine organisms. One thing I want to point out here is the marine ray fin fishes, the class Actinopterygii, which I highlighted earlier, that basically just 8 years ago, there was almost no data available. And so this was really an important paper because it became a real charge to the scientific community globally. And this has rapidly changed. And Trevor was able to capitalize on that. I forgot with Maya, but I don't want to forget now. So Trevor assembled a really wonderful team of dedicated undergraduates, almost all from the Marine Science Department, to work on this research with him. This is a picture of us working in the fall of 2020, and most all of this work, as Trevor said, in terms of challenges, was done, during the height of COVID, and was done this way via Zoom. So as I mentioned, the methods are very similar to what Maya did, except for what he did is he grabbed published data that had been compiled by fishbase.org for 5 ecologically relevant traits: body size, minimum population doubling time, this is how fast the species doubles, habitat tiering, euryhaline status, this is basically if they are moving into brackish or fresh water, and their trophic level. And then again, match these species to their IUCN red list extinction risk categories for almost 10,000 species. So this, right, is just 2014. You could just see it in Doug Macaulay's paper that basically nobody had done anything. Here it is 8 years later, and we have almost 10,000 of those 15,000 species, right? So this is pretty amazing how swiftly we as a conservation community are responding and doing urgently needed work. These methods are almost identical to Maya, so I'm going to skip over them. Really interesting results here. First, I want to highlight the body size results. If you look at this figure right here, here's the histogram, threatened versus non-threatened. In the marine environment, there tend to be kind of fewer threatened species, but about 22% or so in total are threatened. And if you look at the percent of species that are threatened in each of these body size bin categories, what you see here is that big species tend to be threatened, and a little bit the smallest species tend to be threatened, but species that are kind of average size fishes tend to not be threatened. And you can see this here really clearly, especially for critically endangered fishes. Critically endangered fishes are on average really big fishes. This probably doesn't shock anybody, right? If you're really big, and especially if you're a tasty fish, you're in trouble, right? This just blew me away, and this is such a cool figure, at least in my humble opinion, that Trevor generated from these data. What this is here is showing non-threatened versus vulnerable versus endangered versus critically endangered for their basic habitat affinity. So those that are marine and brackish in pink, marine brackish and freshwater in purple, and marine only in blue. And what you see here is that as these species move into freshwater, and you see here in this kind of purple, marine brackish freshwater, the ones that are critically endangered tend to be ones that are diadromous. They're moving between marine environments through estuaries up into freshwater environments. That puts you at very high risk of extinction. And overall here, the signal most strong for the 5 kind of ecological trait categories that we picked are body size and general habitat affinity. So larger body size and or diadromous life histories are generally associated with elevated extinction risk, and especially if you're both. If you're big and you're moving up from marine environments through estuaries into freshwater, you tend to be really highly endangered. He also tallied the threat types and found that the top 5 drivers of risk are harvesting, then pollution, then development, top 4, then climate change. This probably doesn't surprise anybody, right? That harvesting is the big one. And there's some solace in that, I think, right? That harvesting, this is something that we can regulate, that we can change how we go about harvest. We can change, our behavior. But this is, the number 1 threat type for marine ray fin fishes. Climate change here, just to point out, is in the top 4. And that's what this text here says. Again, just like we saw for terrestrial vertebrates, this is same for the marine ray fin fishes, which are by far the most diverse group of marine vertebrates. That the threatened species have on average a greater number of threat types that they're facing than the non-threatened. Again, it's a death by 1,000 cuts scenario. And these results point to the need to swiftly address the extinction risks of larger and or diadromous fishes, and that understanding this rank order of the threat types and the role of the combined drivers of biodiversity loss are necessary if we're to curtail rapid alteration of global marine ecosystems. Okay, so lastly, I asked Trevor, what are your career goals and how has TCBES contributed to those or not? And he said, I'm seeking out to begin a PhD program next fall, focusing on modern extinction with the long-term goal of becoming a professor. TCBES and the Knope lab both helped me transition into the field of conservation from social science and gave me the skills necessary to feel comfortable conducting research completely independently, which would be critical for succeeding in a PhD program. And then I asked them all, do you have any advice for the current TCBES master's students? And Becca said, trust your intuition. Ask for help when you need it and talk to as many people as you can. Jeff said, make personal time for yourself. Take advantage of opportunities to help fellow students, faculty, or land managers in the field. You may get to learn new things about something you may or may not have a chance to work with again, and visit some amazing places and meet some great people. Maya said, take advantage of the wide range of professionals involved in the TCBES program. The TCBES program provides a rare opportunity to interact with university researchers, state and federal researchers, state, federal land management, and more. And Trevor said this really depends on whether you're in the internship or the thesis track. And I think that an internship student would be better able to give advice for that track. He says for the thesis, with the way it's structured in TCBES, it's good to think of it as a mini PhD. In some ways, this makes it more manageable than a PhD. The timeline is shorter, only 1 paper at most is expected. The research scope may be smaller, etc. However, it's still going to be a workload and rigor similar to a PhD. So be prepared to have and develop the skills you would need in a PhD program, ability to conduct research independently, take ownership of the project, become competent in all steps of the scientific process, manage time efficiently with little oversight, and be able to plan and achieve goals years into the future, manage your data, do the analyses, etc. So kind of the whole enchilada. So in conclusion here, I think in terms of their research projects, we can safely say that sea surface temperatures in Hawaii are rising at an alarming rate, and this is coupled to dramatic change in intertidal ecological communities from Becca's work. The DNA barcodes, as they are currently proposed, fail to differentiate native Hawaiian flowering plant species, that should say flowering plant species. Preliminary evidence suggests that island endemic mushrooms support the theory of island biogeography, both of these from Jeff's work with Kira. And terrestrial vertebrates globally face high levels of extinction risk that is strongly ecologically selective from Maya's findings, and that marine ray fin fishes also globally face increasingly high levels of extinction risk that is strongly ecologically selective from Trevor's findings. And then in terms of research process conclusions, creativity and flexibility came up. That also came up in your chat. Hard work and perseverance came up as a theme, both in their work and in the chat. Teamwork, right? This is really important. It is really a team effort. get help when you need it. And this is part of teamwork, right? Is reaching out when you really need help. And to trust yourself, be organized and be time efficient, but have fun and take care of your whole self, right? That was something that, that came up time and again. I just want to end here by showing a picture of myself 20 years ago doing my master's thesis. That's me in the bottom at one of my field sites. This is Punta Banda, Baja California, where I got to spend a lot of time. And I think, it's really important that you do, as the last bullet point here is you also have some fun with it and really take care of yourself as you do your research. I will say that 2.5 years into my research, my master's program, I scrapped my original project and switched gears. And this comes to the creativity and flexibility part, but it took me 4 years to finish because of that. But I was really, happy that I was able to figure out something different that was related. Yeah, it allowed me to finish and publish those results. With that, I leave you with Rainbow Falls, and I thank you very much. I just want to remind everybody who hasn't, go vote, vote, vote tomorrow if you haven't already done it in advance. And I know we're close to the end here, but if there's any questions, that would be great. And of course, I'm happy to answer emails or sidebar with people at another time via Zoom.